All right, good afternoon, everybody. Thanks for joining us today. We always have a few folks continue to hop on in the first 5 minutes or so, but we'll go ahead and get started. And we've got some great content today, so I don't want to take up too much time in the beginning. Just want to say welcome. And of course, today's agenda, we are getting an update on the Brazilian pepper tree biological control situation from Dr. Carrie Montier. I've really been looking forward to this. As you know, we just started doing the releases of the first insect, I guess, right at about a year ago now. So that was really exciting, and I'm looking forward to hearing what's been going on with that. And then we also have an additional update on a new Weed Science Certificate program from UF with Dr. Candace Prince. So looking forward to learning more about that as well. Just a quick note, if you have any questions during Dr. Montier's presentation, please type them in the chat box, and we will, at the end of the presentation, we will circle back and make sure we capture any thoughts or questions you may have. So with that, I am going to turn it over to Dr. Montier. And let's see, I'm going to mute myself, and then Dr. Montier, you should have the controls.
Okay, thanks, Emily. I'm happy to be here today and happy to give you all an update on what we've been doing. As Emily's right, we have been releasing these insects for almost exactly a year. And so give you a little bit of an update on what we found so far. And there we go. Okay, everyone on this call likely knows everything there is to know about Brazilian pepper tree, but just in case, here's a little bit of some background. So Brazilian pepper tree is, the scientific name is Chinus terabithifolia. It is native to South America, mostly Brazil. We like to keep our common names very easy to remember. This is a perennial tree that occurs in coastal and open areas in both Brazil as well as the invaded range here in the United States. So here, this blue line shows the range where it inhabits areas in South America. Brazilian pepper tree was introduced intentionally into Florida in the late 1800s. It was actually introduced twice, once into Punta Gorda and once into Miami. It now basically clobbers the entire peninsular Florida. It covers, last estimate was nearly 280,000 hectares. It invades both disturbed and natural sites, which is why it is so problematic, and it can displace native vegetation. One of the individuals who introduced this plant into Florida was Dr. Geo Stone, and he is a physician that introduced Brazilian pepper tree into Punta Gorda, and he is actually on record as saying it's a very pretty bush and it ought to be in every garden in Florida. I think that maybe it inhabits more than just every garden in Florida at this point, and I think that if anybody who could talk to Dr. Geo Stone in the beyond, he would be very upset with what this plant has done to the state at this point. We've gone one too far. Most people on this call are going to know precisely how we manage invasive plant species. There's many different tools in our weed control toolbox. So we have cultural controls like mulches, weed barriers, drawdowns of lakes, mechanical controls such as fire, mowing, physically disrupting the plants. Chemical controls are always very popular. And then we have biological control, which is what I will concentrate on talking about today. In biological control, it's the use of natural enemies to reduce the density or the occurrence of a pest species. In classical biological control, so this is when you're going to the area to where these plants are from and bringing in something new, something exotic into the invaded range, it will not eradicate a pest. It is just going to reduce the density to a more tolerable level. So I cannot stress that enough. If we get a super successful biological control program of Brazilian pepper tree, we will always have Brazilian pepper tree on the landscape. It is just hopefully going to be at it's going to be at less dense and more tolerable levels. This is a pond in Australia that is covered with salvinia. So this is before any control methods were introduced into this pond. 3 years after the addition of adding the salvinia weevils, this is what that pond looks like. Now, this is a pretty spectacular result. I would be very happy if this was my pond. Not all biological control programs produce this type of results. Sometimes we get lucky and sometimes our insects are super successful and we can brag about this level of control. It doesn't always happen that way, but this is what we strive for. Much reduced density of these species. Now, the theory behind classical biological control works on the enemy release hypothesis. So the enemy release hypothesis states that in their native range, so in Brazil, Brazilian peppertree occurs with its co-evolved natural enemies. So our little dapper yet problematic herbivore here. Now, having to defend itself against these herbivores comes at a cost because it uses resources to defend yourself. And so Brazilian peppertree in Brazil can't be as very good of a competitor because it's having to use resources to defend itself. Now, in the invaded range, these pest species are often introduced without these co-evolved natural enemies. This frees up many resources because the plants no longer have to defend themselves in the invaded range. This frees up resources for increased competition. And the things that these plants are competing with are our native plant species, which are over here on the left, defending themselves against their co-evolved natural enemies. So what classical biological control does is go back to the native range of these plant species, bring back arthropods or insects that we're interested in and think that might do a good job controlling them, test them to make sure that they will be host specific and safe to release, and then reunite these natural enemies in the invaded range. And this is going to return our invasive pest to this scenario over here on the left. And it basically levels the playing field for our native species. Classical biological control has a lot of positives associated with it. It's safe because it's host specific. We spend many, many years inside containment labs to make sure that these insects and arthropods will only eat and reproduce on our target weeds. There's an extremely high return on investment once these insects or other arthropods are out on the landscape because they're self-perpetuating. Insects make more insects. They also move, so they spread into new areas on their own, oftentimes without human input, which means basically once we have them out on the landscape, they do all the work for us. And so you have an extremely high return on investment. This is also environmentally friendly because they are safe and host specific, as often they can reduce pesticide use in areas where these agents are established. Now, with any control tactic, we do have negatives. Biological control is no different. The negatives associated with biological control is that there's an extremely high upfront cost. It costs a lot of money to go to these foreign places and to search for these insects and other natural enemies. The labs that we have to do these studies in are also very expensive. They're designed to keep these agents inside these labs so that they don't get out and cause problems until we know for sure they're safe. It's slow, both with the development of the program, so finding insects and getting them tested to make sure they're safe, as well as slow as once we release the insects. And so we release the insects out on the landscape. It would be great if you could see reduction in density of our pest in 6 months, but oftentimes it takes years, several years, and sometimes even longer than that to be able to see any actual reduction in the density. As I mentioned before, it will not eradicate the pest, it's just going to lower the density. And sometimes the agents don't stick. So sometimes there's abiotic or biotic factors that won't allow our agents to survive or thrive in the new environment. Okay, with Brazilian peppertree, we currently have a USDA APHIS approval to release 2 insects. So first we have Thrips, which is a Thrips species. And the second is Calophyllata forceps. This is a leaf-galling species. Both of these species feed on the young growing tips of the plant. We're going to start with Calophyllata forceps and tell you a little bit about these insects. Well, Calophyllata forceps comes from Bahia, Brazil. So where this star is, this is in northeastern Brazil. So this is a subtropical area. These insects are very small. They're a little less than 1 millimeter long. They produce these pit galls on the surface of the leaves. So here you see a close-up over here on the left, and this is a high level of infestation on the right. The development from egg to adult takes about 40 to 45 days. So here you have your adults that are laying their eggs on the midribs and margins of the leaves. You see the very small eggs here. These eggs hatch, the crawlers then crawl to the top surface of the leaf and try to initiate galling. Basically, they stick their mouth parts into the leaf, and that's where they stay through 5 nymphal instars. So here is the last stage. This is a 5th instar nymph. The entire time they're feeding, and this is acting as a nutrient sink to the plant. The nymphs then emerge as an adults, and the cycle starts over again. So that takes about 40 to 45 days, dependent upon temperature. In our host specificity studies, we found that Calophyllata forceps will lay eggs on 7 different plant species. All of these plant species are in the Anacardiaceae. So in here in the blue, you have Chinus terbiifolia, or Brazilian peppertree. In our orange, this is Chinus molly. This is Peruvian peppertree, and also an invasive in California. And this is Chinus polygamous. This is another congener. Now, you have down here, these are some Toxicodendron radicans. That is poison ivy. This is a native plant, but I don't know if anybody would be horribly upset if we reduced poison ivy, but then we have pistachios here, and then Rhus copalinum over here on the right. So we do see that we get eggs on these 7 species. We do have significantly more eggs on Brazilian peppertree and other Chinus congeners. Now, none of these Chinus congeners are actually native to the US. Now, if this is where our testing stopped, we might have some reservations about releasing this insect because we are getting egg lay on 6 other plant species. However, when you follow those, we found that the eggs either dried and did not hatch, but if the eggs did hatch, nymphs tried to initiate galling and either died, and they all died on non-target plant species. So down here on the bottom right, you have Brazilian peppertree. So this is after 30 days, nice, healthy 5th instar nymphs. Chinus molly, this is Peruvian peppertree again. This is what the nymphs look like after 30 days. All of these nymphs are dead. They did not complete development at all. This is the most closely related plant species that occurs in the US. Over here on the bottom left is pistachio. This is after 16 days, and you see here that we have these dead nymphs. Now, these nymphs did not complete development, and they did not negatively impact the growth of these pistachio trees. So the only plant species that these Calophylla will feed and reproduce on is Brazilian peppertree, so they're extremely host specific. Moving on to damage. Damage on these trees can produce yellowing, of course. You can get deformed leaves, and in severe infestations, you do get leaf drop. With the leaves being the powerhouses of the plant, you get a lot of deformed and yellow leaves and leaf drop. You're going to impact growth of that plant species. So these are a couple studies that were done right before I arrived at UF. Over here on the left, you have proportion of missing leaves. So our blue bars are our control plants, so plants that have no insects, and our orange bars are our plants that have insects. And you see there's significantly more missing leaves on our plants that have Calophylla associated with the plant. Over here, we have an increase in plant height. So we have our blue bar here with no insects and our orange bar with insects. So our plants that have insects were significantly shorter You can see over here on the right, after our caliphia was held on these plants. So they are decreasing the growth of these plants. So the verdict on caliphia is that they are very host specific and they are damaging to Brazilian pepper tree. So as far as this insect goes, that's the end of the good news. So these insects are what I like to call my Primadonnas of biological control agents for Brazilian pepper tree. They are very, very, very hard to rear. They're very fussy. So we have these two ladies here, Carolina over on the left and Kelly over on the right, have been trying to rear these guys inside our containment lab. These insects don't like artificial light, so they have to be in a greenhouse. You can't have them in growth chambers. They don't lay eggs on leaves that are touching the side of their cage. They also won't lay eggs on plants that have a greenhouse pest, such as aphids or thrips or something. And so they're very, very, very picky and very hard to rear in large numbers. As you can imagine, we have to have large numbers in order to be able to release them out onto the landscape. So Kelly over here is looking very triumphant. I'm hoping in the near future she can look triumphant again with the rearing of these guys. But they are very, very difficult. On top of the fact that they are so picky to be reared, they also elicit what's called a hypersensitive response in the plant. So over on the left, this is what we call susceptible plant, which basically means that the plant is susceptible to galling. So that means the insects can lay eggs and the nymphs can grow and come out as adults. Reproduction works just fine. Sometimes we have hypersensitive plants. So the laying of the eggs and the initiation of the galls elicits a response in the plant that will actually kill the nymph. And so all of these brown spots are necrotic spots on the surface of the leaves. So this is a plant response to gall initiation. So this kills the nymph, and I'm sure it damages the plant as well, but we don't get any insects off of hypersensitive plants. And so when I first started at UF, we had probably about maybe 40% hypersensitive plants. And now we definitely have an increase in this hypersensitive response. We're somewhere between 50 and 60% hypersensitive plants. So you get a lot of plants that are hypersensitive, you're basically just killing off your colony. And that's precisely what has happened, is that we have not been able to maintain the colony. So our colony died a few months ago. As everybody knows, we have this wonderful pandemic going on right now. And so we can't travel anywhere. We definitely can't travel to Brazil, which is where we would need to go to get more of these insects. So we have to travel to Brazil to get caliphile out of forceps in the summer months. So down in Brazil, the best collecting for this species is November to January. So we're hoping that by this time frame, we will be able to travel, but it is completely unknown at this point. If we are able to travel down in that time period in order to bring back these insects, we would have to establish a colony containment, which is going to take about 2 to 3 months, because these insects don't really do well in the winter here because of the photo period. And so it takes quite some time to get these guys up and running when we get them back in. Once we get the colony up and running, they have to be cleared by DPI to make sure that the colony is pure, meaning we only have one species of caliphia, and that it is not harboring any plant pathogens. So if all of these things align and we get down to Brazil, we get the colony up and running, and it's cleared by DPI, we'll be doing releases mid-2021. But that's best case scenario. But that's the end of the bad news. Let's get talking about the thrips, which is where we have much more exciting things to report right now. So this is the Brazilian pepper tree thrips, or Pseudophilothripс itchini. It's what has a face only a mother could love. So here's the adult over on the left. You have our immatures here, the yellow ones, and this is an adult here. So these guys are super charismatic. I think if you're an entomologist, at least we think so. They have these really neat defense mechanisms where they curl their abdomen up and look like tiny scorpions. These guys are extremely damaging. So once we put a plant into a colony, you get out a dead plant. So these are on very small plants, not large plants, of course, because they have to be in cages, but you can see all these dead tips here. Okay, the Thrips species take about 30 to 35 days in order to complete their life stage. The adults lay eggs on the new growing shoots of the plant. We have 2 larval instars, as you will see here. Both of those feed, and they feed on the tender new shoots of the plant. We have 3 pupal stages. Pupation occurs in the soil, and then they emerge as the adult. The adult also feeds on the growing tips of the plant. The Thrips were tested on 127 different plant taxa, so they were very thoroughly tested. These plant taxa occurred across 45 families and 33 orders. And what we found is that these Thrips will only feed and reproduce on Brazilian peppertree. They will actually feed and reproduce a little on that Peruvian peppertree, but not at a rate that will actually establish a population. So Peruvian peppertree is also another invasive. Now, these Thrips are extremely damaging. This is after a 30-day exposure to Thrips. So we've got in our white bars here, these are control plants, so plants that do not have any Thrips on them. This, we're looking at percentage of dried stems. In the gray, you have the plants that had the Thrips put on them. So directly after herbivory, so this is after a 30-day exposure to the Thrips, we see that the plants that had Thrips had a much higher percentage of dry stems than ones that don't. Now that's good. We like to know that our biological control agents are actually impacting the plant. But what's really exciting here is 2.5 months after recovery. So this is 2.5 months after the Thrips were pulled off of these plants, all of the plants. You have no dried stems in your control plants, and you still have a high percentage of dry stems on your plants that had Thrips. And so not only are these Thrips damaging to the plants when they're actually on the plants, these plants do not recover very well once these insects are removed. So this is extremely exciting because we have a lot of Brazilian pepper on our landscape. Okay, the verdict on the Thrips. They are host specific. They only feed and reproduce on Brazilian peppertree. They're extremely damaging, as you saw in the previous slide, that even these plants don't recover very well after the Thrips have been removed. They were approved for release a year ago, just like Califia was, and releases began back in late June of 2019. So far, we have released about 139,000 of these Brazilian peppertree Thrips across the state. So this is both my lab as well as the USDA ARS Invasive Plant Research Lab down in Fort Lauderdale. So we are working together on this. FDAC's DPI is actually joining the team very soon to help us get more of these insects out on the landscape more quickly. So far, we have released these insects across 15 counties. We have looked at whether these insects can be released a single time. So you take Thrips out once, leave them there, and they're good to go, or if you need to release them multiple times. We have also looked at release sizes. So we've looked at releasing Thrips in 100, 300, 900, and 2,000 Thrips groups in order to find out precisely how many Thrips it takes to get these guys established on the landscape. We don't want to concentrate on releasing 100 Thrips every single release and then them not established. It's a waste of resources. On the other side of that coin, we also don't want to take out 2,000 Thrips into a particular area if 900 Thrips would do. And so right now we're trying to figure that out. And what we have figured out so far is that releases under 300 Thrips do not work. We do think that these guys are being predated very quickly, and so smaller releases aren't working. These are single uncaged releases. Like I said, predation super high. Ants and spiders love these thrips. We have seen them literally getting taken away in just moments after release. And so we have started to try to do cage releases, which you can see here. So this is a sleeve cage. What these sleeves do is these sleeves will both protect our thrips from predation because it keeps things out of this area, and it also keeps the thrips where we put them. When we are going out and releasing these thrips, when you have 300 thrips that you're releasing, you come back and you have a quarter-mile stretch of Brazilian pepper tree, sometimes it's hard to find them. And so releases of 300 and 900 thrips, we might not be able to find them. They might be there just fine. They just might be in areas we're not looking. And so we're caging them to see if that will make it easier to find them after release. So that's what we're doing currently, is testing cage releases. As you can imagine, we want to be able to get these out to the public. And so we have to be able to figure out how we can get these insects established in a single release. It would be really difficult to get these out to the public, to be able to get them to John Q homeowner, but then have to get thrips again out to the same John Q homeowner. So we want to be able to figure out precisely what we need to do, if it's a cage, if it's not caged, or how many insects it takes to get these out and get these established with one release. So public releases are coming. We're very close to getting the answer to what we have to do to be able to get those out to the public. So once those release strategies are solidified, we will begin to offer these to the public, which I know everybody is waiting for. I get emails and phone calls nearly daily. So we are definitely trying to get these out to the places that need it. And now that we have my lab, USDA, ARS, as well as FDACS DPI, we're hoping that we're going to be able to get these out quickly, but we're still not quite there yet. So a little bit more patience, and we'll be able to do it. In the meantime, we are conducting an economic survey. So it's important to know what people are currently spending on the control of Brazilian pepper tree. And so this is a survey that we have designed to get to that answer. And so this is a really long link. I'm hoping that Emily will be able to help me out and get this link out to the people on this call to, if you all are interested. It's an extremely low time commitment. It takes about 10 minutes to take this survey, but it's definitely going to help us out, because we then can take the data from this survey, and in 5 or 10 years from now, once we have these thrips out on the landscape established, can send it out again, so that we can calculate the effect or the economic impact of these biological control agents. So wait, we got Emily, I think, playing around. So so that's basically it. We've got our economic survey there. Acknowledgements, of course, there's so many people that I have to thank for this, both funding agencies as well as collaborators. We definitely don't do this alone. We have, I work with people down at the USDA lab, I work with people at FDACS, and we also have people at UF-IFAS, people that came before me and did a lot of this research before. And I can't thank enough the extension agents that have helped us get these insects out on the landscape right now. And here are my contact information. If you would like more information, we also have social media pages. So we have both a Twitter as well as Facebook. That's likely going to be one of the first places where we announce that we're doing public releases. And so pay attention to those, because if you want these insects, we're trying to get them out as quickly as possible. And with that, I'll take any questions that you all have.
Great, thank you so much. Yeah, sorry about that there at the end. I was trying to find that link. So I put the link to that survey in the chat box here. So folks can click on it straight out of the chat box. And we've had some questions coming in, so I'm gonna go back and start from the beginning. So going back to the Calophyia, with that one, do you anticipate them having any natural enemies in the US once released?
Oh, of course. I'm just like the thrips. All of these biological control agents become integrated into the ecosystem. And so there's definitely going to be things that will be predating on them.
Okay. And where do you intend to do the releases of the Calophylla? Kind of in the same areas, just anywhere that Brazilian pepper is?
Yeah. Well, so the Brazilian pepper tree thrips, if you all remember, pupate in the soil. Calophyia, the entire life cycle occurs on the plant, so on the leaves of the plant. And so Brazilian pepper tree can occur with wet feet in many areas. And so we think that the thrips are not going to be as effective in those areas because they pupate in the soil. And so we will likely start releasing Calophyia in areas where the thrips might not have as much of an impact. So mangroves, the Everglades, places where Brazilian pepper tree has wet feet.
That's really interesting. Thank you. So someone just asked to clarify where the thrips themselves are native to.
So they're native to Ouro Preto, Brazil. So that's kind of central on the East Coast, but central Brazil.
Awesome. So just a comment here. I know FDAX works with local extension office to distribute air potato beetle. It would be great to know the timeline of distribution so we could plan to host a class in conjunction. We're always looking for ways to teach people in conjunction with free giveaways. So I know you mentioned coming soon. Do you guys have a target, or where do you land on that?
We don't have a target date just yet. So we just started cage releases probably a couple months ago. And so as the data comes in for that, we'll be able to get a better timeline. So we're just waiting on those data to come in. So like I said before, in order to release these to the public, we need to know precisely how to release them so that we can get them established at one time. I would hate for somebody to come into an extension office, go home with 100 thrips and then not see anything.
Absolutely. So. Thank you. And I will second that. If you're not currently following these social media pages and you are active on social media, this lab is really good about providing updates and talking about their research. So a great way to keep up with what they're doing. Have you done any releases in northern counties yet? So Duval, St. John's, and even in Nassau, of course, we're starting to see Brazilian pepper.
So we've done releases in Brevard County, which is I think the most northern county. So on the East Coast and on the West Coast, we have gotten up to Pasco County.
Great. Has there been any, so with these northernmost releases, has there been any notice of how cold tolerant these thrips might be?
The thrips are basically have the same thermal threshold as the plant does. And so we expect these thrips to be effective all the way up into southern Georgia on the coast.
Oh, great. Thank you. Have you gotten any negative feedback from beekeepers?
Oh, of course, of course. Most biological control programs of flowering plants are going to have negative comments from beekeepers. And I don't think that's necessarily because we're at odds with the beekeepers. I just think that it's an education thing. So as I said before, biological control will never, classical biological control will never eradicate a pest. So it's just going to reduce the density. And so Brazilian pepper tree is already being controlled out on the landscape, and it's being controlled in very bee unfriendly ways. And so chemicals aren't good for bees. Cutting down Brazilian pepper tree is definitely getting rid of that floral source. because biological control works so slowly and so naturally, you're just going to get a reduction. So it's still going to be on the landscape. And even better than that, we're hopefully going to get a resurgence of some of our native flowering plants so that the bees will have more diverse floral sources.
Great, thank you. And so that was the last of the Zoom chat questions that have come in. I have one question that I'm going to ask, so this will give everyone kind of a last chance. If you have any last minute questions, go ahead and get those typed in. So the question I have is more about biocontrol in general, but as you begin to be able to release these to the private homeowners and private lands, you know, obviously we'll see a resurgence of these questions about biological control in general. And I'm pretty good at answering most of them, but one of the ones I get that I kind of don't know the absolute best answer for is, how do you know it just won't evolve to feed on native plants? That people seem to be convinced that that is the inevitability of releasing these on the landscape. So how do you answer that question?
So in order for evolution to occur, there really needs to be some catalyst for it, right? In the lab, we do two different types of tests. So we do one no choice test. And so these are extremely conservative tests. So this is basically where we take a plant species, a non-target plant species, and we put the biocontrol agent on these plants. There's no other choice. They have to feed on that plant or they die. If they were able to survive on this plant, they would. And so the biological control agents that get approved are usually the ones that don't feed on these in these no choice tests. If we do get feeding on a no choice test, then we go to choice tests. And so say we had pistachio, and so say one of our agents fed on pistachio, and so then we put Brazilian pepper tree and pistachio into a cage, and then look at their choice, because oftentimes these biological control agents will choose overwhelmingly so our target weed. Now, if you're getting a lot of choice on that non-target, we're not going to get this thing approved. And so you are definitely finding the species that can only feed and reproduce on our target plants. And since we will never eradicate our target, there's not really that catalyst to get that evolution to occur, right? Like, you're not really going to be into a point where all the Brazilian pepper tree is gone. And so then all of a sudden you have to be feeding on something else. These insects die as opposed to reproducing and keeping a sustaining population on those non-target species.
Great, thank you so much. One last question. So we talked about the working towards the process to start getting these out to private landowners. As far as what you're currently doing, is there a method for public landowners to request releases, or is that gonna kind of be rolled out at the same time?
Well, so that all depends on funding. So if you are a land manager for public lands, shoot me an email, because that's definitely something that we can explore. So far, we've been funded to release on cattle ranches and Air Force installations. And so that's where a lot of our releases have gone, because that's where the funding has come from. But we do have a little bit of funding to be able to expand that to the public lands. So yeah, definitely shoot me an email if you're a land manager of, you know, some public lands.
So one more question coming in here, and I think you hit on this a little bit before. Are you looking to utilize the same release sites for the Califia that you have for the Thrips?
Yeah, not necessarily. We would definitely want to put Califia in wetter areas, but we are aiming to release both of these insects across the entire state, because the more help we can get controlling this plant, the better.
Awesome, great. Well, thank you so much for this presentation. And again, for folks who have any additional questions, definitely follow the lab on social media, and you can reach out to Dr. Mentier as well. So thanks again. And with that, I want to go ahead and introduce Dr. Candace Prince. She is going to share some information on a great new program being offered through UF IFAS. So Candace, you should be ready to take control. Just click on the screen. Thanks.
All right, thanks, Emily. I'm really excited to be talking to you all today about our new Weed Science Graduate Certificate Program. We've been working hard over the last year to put this program together, and it's officially launching this coming fall semester. So I'm grateful for the opportunity to talk to you all about it today. So what is an online certificate program? It's really a continuing education and professional development opportunity. It is fewer courses and requirements than a degree program. So this certificate is 3 courses or 9 credits compared to a master's degree that's 30 credits or a PhD program that would be 90 credits. And then it has an online final exam. And it's a fully online program. So there's no requirement to come to Gainesville. For many of the classes that are in the certificate program, the lectures are pre-recorded. So you can go at your own pace each week and fit it around your work schedule. And so the Weed Science Certificate Program is a graduate level certificate within the agronomy department at the University of Florida. And it was developed to provide training on weed ecology and plant interactions, as well as management techniques and environmental considerations for invasive species. And so we really developed this for natural resource managers, people who are working in invasive plant management, but maybe their educational background is in wildlife ecology or biology, and they're looking to learn more about plants, agricultural managers who have to deal with agronomic weeds, government employees, industry personnel, extension agents who are involved with invasive plant efforts, and also for students who have been thinking about going to grad school, but maybe want to dip their toes in the water a little bit and do some classes before fully jumping into a master's degree or a PhD program. And so as part of this program, you'll learn to describe the biology, physiology, and impacts of invasive species, learn how to use appropriate and environmentally sound management methods, and understand the science behind why we use different management techniques. There's one required core course for the program, which is integrated weed management that's offered every fall. And this is a really good course that goes over different types of management activities, what makes a species weedy or invasive, as well as some weed identification. And then we have a list of 5 electives that you can choose any 2 from. So we have a couple of courses that really focus on pesticides and herbicides. We've got Principles of Pesticides that gives a good overview of what a pesticide is, how to use it. Plant-herbicide interactions, which really dives into how different herbicides work on the plants on a physiological level. So what is it that makes glyphosate act differently than, say, a Mazapure when you're applying it to a plant? We've got one course that's offered every other summer that specifically talks about conservation and restoration. So how do you restore an environment after you remove an invasive species? And then we have a course that focuses specifically on aquatic plant management. So aquatic invasive species identification, how to manage plants in an environmentally responsible way in aquatic environments. And then a course that focuses on invasive plant management in upland systems. So for requirements for admission, there's no GRE requirement for the program, so you don't have to take the GRE. We do require that you have a bachelor's degree. So a bachelor's of science is preferable, or a bachelor's of arts degree, as long as you also have some experience working in plant management. And we do require that you have 1 prior course in plant science or experience working with plants. So this could be a course in botany, plant physiology, agronomy. And the reason why we have this requirement is because a lot of the courses, specifically plant herbicide interactions, the aquatic course, go into a lot of plant physiology aspects. So we just want to make sure students are prepared for that. If you are interested in taking the program, but you don't have a background in plant science, we do have an undergraduate plant science course that you can take online over the summer, which would prepare you for the program. So this is a 9-credit program or 3 courses. You can complete the coursework over several years. There's no time limit. So you could take 1 course this year, you take a course a year later or 2 years later if you get busy. And if you do decide to go towards a master's degree or a PhD, you can put those credits toward that program as well. We do require that you have an overall GPA of 3.0 in order to complete the program. And there's also a final exam that you'll have to take, which is about 10 questions from each of the courses that you enroll in. For the cost, it is about $550 per credit hour. That works out to about $1,600 per class. That's a flat rate, whether you're in state or out of state, and it includes all tuition and fees. If you are a Florida State employee, so if you work for FWC, for example, you may qualify for a state tuition waiver or a reimbursement program, so your costs would be waived. And so it's an online application. We just need your transcripts and your resume. And we ask that you apply 1 month prior to the start of the semester that you want to enroll in. So the deadline is pretty close for fall. If you're interested in applying for next spring, we just would require that you have your application in by about November, end of November. So if you have any questions on this program or you want more information, feel free to contact me. I've got my email address listed here as well as my phone number. If you have any questions that are specifically about navigating the application process in our online system, you can contact our academic program coordinator, Cynthia Height. I've got her information here as well. She's awesome and would be very helpful. And with that, I'll take any questions.
Awesome, thank you so much. Are there any questions related to the course? I will go ahead and advance to, I just have a very quick update about the calls. But if you have any questions, go ahead and type those in the chat box now. Thank you so much, Candace, for sharing that with us.
Thank you.
So really just a small set of reminders today. Wanted to share the rest of our 2020 call schedule with you. So next month, we will hear about a really cool coordination program going on in the Panhandle that has been a partnership between Six Rivers and the Longleaf Alliance to do some regional Kogan grass coordination. So really excited to just hear about how they kind of got that together and created this position to do this work. In September, we have the mechanical harvesting of aquatic invasive plants with Dr. James Leary from UF. And this is some research that was kind of born out of the whole plant pause situation, the aquatic plant treatment pause, and the desire to put some more research into just all the tools we have in our tool shed for addressing aquatic invasive plants. So looking forward to hearing that as well. And then in October, we'll be hosting Dr. Melissa Miller, who they recently released a study on the parasite, the novel parasite that has come in with the Burmese pythons and is affecting our native snakes beyond the range of the python itself. So really cool study if you haven't seen that yet, and looking forward to learning more about that with Melissa. So that is our 2020 call schedule. And then just a reminder that that past calls are all available online on our website. It takes a little less than a week typically to get them up. So if you're looking for this call by midweek next week, it should be available. And please follow the Florida Invasive Species Partnership on all our social medias. We've been creating some new products that hopefully you guys can use and share in your own program. So with that, Candace, we have had a few questions come in. Okay, could you post a link to the graduate classes? Candace, do you have a link?
I don't have a link handy with the description of the courses, but I have a document that I can send you, Emily, if you would be able to maybe send that out after the call.
Send that to me and I'll send it. I'll follow up with the CISMA listserv and share that information.
Okay.
Is there so one question, state employees may have tuition waivers, but what about municipality staff? Is there anything you're aware of for them?
That's a good question, and I'm not sure the answer. So a lot of it is going to be specific to who your particular employer is. And I know even within the state agency, sometimes it works a little bit differently, whether it's a tuition waiver or if it's a reimbursement program. So I would speak to your supervisor and see if there are any programs in place. I know there have been some federal agencies that have been able to get the tuition covered by their employers as well. So I would encourage you to check with the specific group that you work with and see if they have any programs in place.
Great, thank you so much. So with that, that's all we have for today. I just want to, again, shout out to Dr. Mintier and Dr. Prince for joining us today and providing this information. I hope everyone has a great rest of your week, and we'll talk to you next month.